Good evening, everyone, and welcome to the first of this year's Oxford Botanic Garden and Arboretum Autumn Science Lectures, sponsored by Plants, People, Planet. My name is Simon Hiscock, Director of the Oxford Botanic Garden and Arboretum, and it's my great pleasure to introduce tonight's speaker. But before I do this, however, I need to go through a little bit of housekeeping with you. First of all, this lecture is being recorded, but your camera and mic are turned off, so you're not being recorded. If you would like to ask a question to the speaker, then please use the Q&A function at any time, and we'll collate these questions and I will pose them to the speaker at the end of the talk. If you need to contact us about anything else, for instance, any technical problems, technical issues, please use the chat function and one of the team will get back to you. The recording will be available for attendees after the event, and we will be sharing details about how to access this via the emails that you used to register for tonight's lecture. And now to tonight's speaker. I am delighted to welcome Professor Sophien Kamoun from the Sainsbury Laboratory in Norwich, where he is research group leader and Professor of Biology at the University of Norwich. His research group studies plant pathogen coevolution and how it impacts mechanisms of virulence and immunity, particularly the interactions between plants and filamentous pathogens, most notably the fungus-like microbe Phytophthora infestans, which was responsible for the Irish potato famine, and the rice and wheat blast fungus Magnaportha oryzae. His research uses genomics and molecular biology to gain insights into the biology and evolution of these plant pathogens and the plant immune systems that have evolved to resist them. He has published over 150 papers and has received numerous awards, including the Syngenta Award, the Noel Keane Award from the American Phytopathological Society, and more recently, the Linnaean Medal from the Linnaean Society of London. In 2015, he was elected a member of the European Molecular Biology Organization, EMBO, and in 2018 as a Fellow of the Royal Society. Sophien, it's a great privilege to have you here tonight to launch the 2022 Autumn Science Lecture Series with your talk, Keeping Up with Plant Killers. Over to you, Sophien. Thank you.
Thank you, Simon. Thank you for the kind introduction and also the invitation. It's my honor to launch this series, which looks absolutely fantastic. I have to say, it's really wonderful lineup of speakers. So let me get my slides here going. I hope everybody can see them clearly. What we'll do tonight is we'll be talking about plant killers and plant immunity. So as Simon told you, I'm a plant pathologist at here in Norwich at the Sainsbury Lab, and I'm interested in how these various microbes and pests can infect and colonize your plants. And anyone who has a garden can testify to that. Interestingly, the United Nations in 2020 declared the year as the International Year of Plant Health. Sadly, of course, there was another pandemic that took over and distracted us, but we should not forget that there are many pandemics that affect crops like wheat. This is a photo from our colleagues in Bangladesh who are suffering from a pandemic caused by a disease called wheat blast, which is threatening the wheat crop, which is about 20–25% of all the world's calories come from that one crop. So we should not neglect these plant pathogens, and really they have a lot of impact comparable to what we've seen with the other pandemics. But before we get to that, I'm going to take you through a little tour of these plant pathogens, and you realize why I actually admire them. I suspect many of you here, because the lecture is associated with the Botanic Gardens, many of you here are natural history buffs, and me too. I am actually. I grew up. My journey to biology came through natural history and through my fascination as a teenager of the natural world. And what I'm going to try to do tonight is to really navigate through how awesome these pathogens can be. Sadly, they are awesome, but also how destructive they are, and how we can deal with that, and how we can really respond to these pandemics, these plant pathogen pandemics. So let's start in the United States. This is a big country, and right in the middle of that, you can see the Rocky Mountains, which really split the country into, I suspect a number of you have traveled there and have seen how beautiful the nature is. My partner, Saskia Hornaut, who sadly is on cooking duty tonight because I'm giving this lecture, I have a good excuse for not cooking. But Saskia and I were in this part of the US last summer, and we were in Wyoming and visited the Grand Teton National Park. And I'm going to show you a very impromptu video we made last summer when we were in Grand Teton. This is very high elevation. These are alpine meadows. No, 3,000 meter, close to 3,000 meter in altitude, maybe about between 2,500 and 3,000. And we saw something we love and study. And actually, Saskia is the famous word expert in this. And we decided to make this video with our iPhone. So let's watch this. Hey guys, you know what? We're in Grand Teton National Park. Look at all the green. This is, plant lover place, I tell you. But you know what we found? We found something really cool. So Saskia is going to tell you about that. Hey Saskia, what did we find?
Look at this.
What's that?
That's Bushera.
Oh yeah, that's Bushera. That looks like a nice little flower.
Beautiful, healthy flower. A purple, beautiful flower.
Let's see if I can focus on it.
Yes.
Yeah, anyways, you can see that, right? I will post some photos, but that is Bushera, right? Probably Bushera stricta, I guess.
Healthy plant.
Healthy plant. What do you mean healthy? What else?
Right, so let's look at an infected plant.
An infected plant? Yeah. Infected with what? With the phytoplasma?
Fungus.
What?
Here.
Where is it? Oh, it's hiding there. Oh, look at that.
It's like...
That's the same plant.
It's a zombie plant.
But it's infected with the fungus, with the rust. Buccinia monoica, with the rust fungus.
Here. Look.
It doesn't look at all like the other plant. So what is this? What are we going to call it?
A zombie plant.
A zombie plant. Are there more of these? Did you see more of these?
Yes. Over here.
Over here. Let's find out. Saskia, the zombie plant expert, is going to show us more zombie plants in the wild. Oh yeah, look at that. Yeah. It is. Let me focus on it. That's definitely an infected plant. Look right next to it.
And healthy one.
Yeah, there's our flower, our brassicaceae, and infected, doing fine. It's a little late in the season, so they're not as spectacular as they normally are. There's more. Oh, there's more. There's more.
Over here.
This is so exciting. These are zombie plants. They're totally taken over by the fungus.
But here is a beautiful one.
And the plant's not going to set seeds, so it's game over as far as plants are concerned.
The beautiful one is over here. There's
There's more of these. There's a healthy guy.
A healthy guy.
There's another healthy guy.
Healthy guy.
Many of them have not been infected. So infection is not 100%, obviously. Otherwise, the poor plant will just be gone. Because this is game over as far as the plant is. Oh, yeah, we have a pretty one here. Oh, this is the prettiest of all. Look at that. Oh, my God, look at that. Oh, that's zombie. It's still yellow. There's still some spores on it. The yellow color is from the fungus.
Look at the flower.
Oh, yeah, I don't know if I can focus on that, but we'll have photos. Yeah, look at that.
There.
Whoa.
That is amazing. And guys, this is like really close to the lodge, Jackson Lake Lodge. Just like, I don't know, half an hour walk from there. And right there, you have this amazing, amazing piece of natural history.
It's there.
And the mountains, oh, look at the mountains. Yeah, look at that, the Tetons are there. So we can join here, natural history, biology, and pleasure. See you, bye.
Okay, I hope you enjoyed that. We did, and this is what these plants look like when they're infected with the fungus. So incredible, isn't it? I mean, the fungus is completely reprogramming the plant and totally transforming it. These are not flowers. You'll be forgiven if you think this is a flower, but these are actually leaves that are now mimicking a flower. We like to call them fake flowers or pseudo flowers. And the fungus does this because the fungus wants to attract insects, to get pollinated, to get essentially have the insect carry its spores from one plant to another, and that's how the fungus can go through its sexual cycle. In fact, if you cover these plants, then the fungus won't be able to reproduce and go on to infect more plants. Incidentally, we studied this at the molecular level and showed that there are, as you can imagine, many molecular pathways that are manipulated by this fungus. There's, for example, the plant volatile pathways are actually shut down. The fungus shuts down the volatile production of the plant and makes its own. So in fact, these fungus flowers, if you like, are very smelly. They smell very strongly, and they're even more attractive to insect than the normal flowers or the original flowers. So I love this because this is a very vivid illustration of what these microbes and pathogens, like the rust fungus, can do to their host, to the plants. And as I mentioned, the fungus is absolutely incredible in its capacity to manipulate the plant. And some of its relatives, some of the relative of the Puccinia monoica, which causes the pseudoflowers, are actually threatening our crops. So this is an example of the rust fungi that infect wheat and other cereal crops, and they're incredible. Their capacity to evolve is absolutely astounding. They're continuously producing new races with new traits. They overcome fungicides, all sorts of things. And another amazing thing besides that is just the sheer number of spores they produce. So if you walk through one of these rust-infected fields, you'll be totally covered with spores, and you can see that cloud of spores behind the tractor. So an incredible capacity to evolve and produce, spawn new mutants and new races, as we've seen also with the coronavirus. The capacity of these parasites and pathogens to evolve is absolutely astounding. They're continuously spawning new pathogen races and acquiring new traits that make them more virulent. So let me switch to another pathogen that's quite formidable. I spent most of my career studying the Irish potato famine pathogen, Phytophthora infestans. This is a fungus-like organism that infects potatoes and has triggered the Irish famine back in the mid-19th century. You can see how destructive this pathogen can be. The plants in the middle are actually immune. They carry a single gene that confers resistance to this pathogen. I'll talk about that later. But the two rows of plants surrounding it don't have that gene and therefore get completely wiped out by this microbe. And as you know, this had a huge impact on human history with what happened in Ireland. Of course, there were many factors, many socioeconomic factors that led to the famine, but the pathogen and the outbreak that it caused at the time, the destruction on the potato crop, triggered a lot of these events. One piece of trivia that always stuck in my mind is that the population of Ireland has not recovered to pre-famine levels. And that's just an incredibly sad and sobering piece of information. Let me show another film. This is an animation made by a former student, Remco Stam, who's now a professor at the University of Kiel in Germany. And he made this time-lapse video of potatoes sprayed with the pathogen, with Phytophthora, which is Greek for plant destroyer or plant killer. And the time-lapse is just over a few days, so basically 4 days, 4 or 5 days maximum. And you can see how the pathogen spores, he sprayed the potatoes with spores of Phytophthora, and then the spores will germinate, they'll penetrate the leaf, and then the pathogen will rapidly spread and grow throughout the potato plant. And as you can see, it wouldn't take very long before the entire plant is totally dead. So it's, again, a really destructive pathogen, and unsurprisingly known as the plant destroyer, which is what Phytophthora really stands for in Greek. Let's skip the fancy music. Oh yeah, so let's zoom in on one of these areas. So this is a more microscopic view of what's happening in a potato leaf when it's infected by the pathogen. A few spores were placed here in the middle of that lesion, and then you can see the Phytophthora labeled in red here. It's expressing a protein that's fluoresces in red. You can see how it's spreading. And you can also see a few things here. If you go here on the left side or the right side, you can see the pathogen is really plowing through a healthy plant tissue. And that's one feature that these pathogens have, and something we've been studying for many years, is the capacity of these pathogens to really suppress plant responses, to be able to establish themselves in the plant tissue and really grow and colonize it before things go awry, if you like, and then you get the necrosis, as you can see here in the center, where then the pathogen would be sporulating and producing more spores and essentially growing on dead tissue. So a pathogen like Phytophthora has this capacity to have both invade healthy plant tissue and suppress plant defenses and various plant responses, manipulate the host, but then ultimately it just kills the plant and feeds on that dead tissue. So one of these questions that we've been studying for many years is, how do these pathogens infect plants? How do they cause disease? And this is a topic that, of course, many plant pathologists and entomologists also have been studying, how these pathogens and pests can colonize the plants. And this is the answer. So without getting into a lot of the details, let's simply think of these pathogens having a bunch of keys. They have a number of keys in their pocket, and these keys can help them unlock various plant processes and modify the host environment, modify the plant environment to essentially shut down responses, shut down defense responses, modify the plants in many ways to basically colonize it. And so we like to call these molecules effectors, which are secreted by all types of pathogens, including, by the way, parasitic plants. So, you know, many species of parasitic plants, and also parasitic plants can also modify the host plants they infect and colonize by secreting these effector molecules. It's a very interesting concept, the effector concept, really. I'm going to expand a little bit on it, because if you think about it, this is an amazing concept, because the genes for these effectors are actually sitting in the parasite or in the pathogen genome. So the parasite is producing a molecule from its own genes that is not functioning inside its own cell, but functioning in the plant cell. And so that's how these pathogens can modify the plant in such a spectacular way. So I showed you the zombie plants. These are the ones that actually Saskia also studies, caused by a bacterium called phytoplasma. And the presence of one single effector, just one single gene, can totally reprogram the development of the plant and turn actually these flowers into leafy tissue, which is good for the microbe. It's enabling it more biomass to grow, and it's also attracting insects, which are essential for vectoring from one plant to another. So Richard Dawkins, is actually described this concept without really talking about effectors. He didn't know about effectors at the time, but he described the concept as in his book, The Extended Phenotype, because essentially what effectors are, they are genes sitting in the parasite, but have their phenotypic expression in the host. And so in a way, this definition taken from Richard Dawkins' book is still valid today. It's a fascinating concept to me because pathogens and parasites evolve so rapidly and have such capacity for biological innovation, it's ironic that some of the most remarkable molecules we know that function inside plant cells are not actually plant molecules, but they come from the parasite and the pathogens. So there are many hormones and many mimics of plant hormones that are produced by microbes. In fact, the first plant hormone, gibberellin, was discovered from fungus. It's not the first plant hormone, but it's the first time gibberellin was discovered from a fungus before it was then later on validated as being a plant hormone. And there are many other really amazing effectors that do all kinds of things to the plant using all kinds of molecular tricks. But I really like this idea that some of the most remarkable molecules that function inside plant cells are actually not from plants, but from these microbes and parasites. So that's one concept, that effectors are the key to the capacity of these pathogens and parasites to infect the plants. So effectors are key to virulence, if you like. Okay, I'm going to take a little parenthesis here and decided to sort of take you on a world tour. So let's travel now to Australia. This is actually our car, Saskia and I, when we were in Australia way back in the 1990s. We drove around and camped and did a lot of insect collecting, actually, and insect studies at the time. In this Australian outback, there is one plant that is absolutely remarkable for very selfish reasons. I would say that this plant, I owe a lot to this plant for my research career. This is a wild tobacco, an Australian tobacco called Nicotiana benthamiana. We like to call it Benthi because we love it, so we had to give it a nickname. And Benthine is an absolutely amazing plant for this particular reason. We can very easily use Benthi leaves to express any gene we're interested in. And we do this using a little trick. As you can see here with this syringe, we are infiltrating the Benthi leaf. And we infiltrate it with a bacterium known as agrobacterium, another plant pathogen. But in this case, a plant pathogen that's useful to us because we can modify it to deliver any gene into the plant, any gene we want into the plant. So that particular leaf, as you can see there, is totally infiltrated with that agrobacterium solution, is now within a couple of days will be expressing our gene of interest. And this has been absolutely fantastic tool for us. So this is a poster that illustrates all the things we can do with Benthi when after we agro-infiltrate it. We can, for example, do cell biology. We can do pathogen assays. We can do biochemistry. There's essentially dozens of ways we can use this system. And what I love about Benthi is that it's because of the speed of these assays, we can really accelerate the research cycle. So generally in my lab, the Friday is known as infiltration day. So most of our scientists will plan their experiments. So they do the infiltration on Friday and they go on their weekend. And then when they're back on Monday or Tuesday, they can analyze the results of that experiment. And very often they'd be able to plan another one for the following Friday. So because the experimental cycle is so short, literally a week, we can test and challenge a lot of different hypotheses and therefore be more creative with our research. So Benthis allow us also to study plant defense and plant immunity. And I'll be getting to that topic now, but this is simply to show you that if we take, for example, a potato gene that is functioning and providing resistance to Phytophthora infestans, the Irish potato famine, then if we take a Benthi leaf that is expressing that gene, we don't get an infection. You see the spores there cannot go on and colonize the leaf. But if that gene is missing, then we get lesions and infections. So it's a wonderful system to study plant pathogen interactions and study plant immunity. that was a little parenthesis. I want to introduce you to Bentley's, a plant we love, another plant we love. But let's go back now to Norfolk. This is a photo we've taken from one of the many walks we did during the COVID lockdown. I'm sure you all experienced the same thing. It forced us to discover Norfolk a little better than we probably would have. And so when you go on this walk, I mean, there's something that's very striking, right? How green everything is. And I spend now, 15 minutes telling you about how pathogen, how great, and how fast they can evolve and all of that. But really, when you go on your walks, most of these plants are absolutely healthy, right? They're just doing fine. They're not infected by any microbe. There's no insect chewing on them. There's no nematode eating their roots. They're just perfectly green and healthy. And indeed, that's an absolute dogma of plant pathology that plant pathologists have noticed many, many years ago, maybe a century ago, that most plants are actually resistant to most pathogens. So disease is actually an exception. Resistance is very common. If you take your average pathogen and throw it on an average plant, it will not infect it. Most pathogens are unable to infect the majority of plants. It's funny because that's in plant pathology, we like to say, look at it from the plant side, that resistance is common and disease is the exception. Our entomologist colleagues, they look at it from another angle. They say almost insects are specialists. They can only infect few plants, but literally it's the same thing, right? It's just the same concept looked at from a different angle. But the key point, whether we're talking about insects, whether we're talking about nematodes, bacteria, viruses, fungi, phytophthora, the reason disease is the exception and resistance is common is because plants have an immune system. And therefore, that immune system makes them resistant to the majority of pathogens and pests that are out there. So let's explore a little bit how pathogens can detect these, how plants can detect these pathogens and have such an effective immune system. Where they do it, because they have receptors, okay? So receptors like these are here shown. I don't want to get too technical, but these are known as NLR immune receptors. Incidentally, we have very similar receptors in the human body. So these immune systems have evolved very early throughout evolution and are present not just in plants, but also in animals and even in microbes like bacteria and fungi. So these receptors are able to intercept and detect these effectors, okay? So as the pathogen is delivering effectors inside the plant cell to modify it, and those effectors are absolutely key to the capacity of the pathogen to cause disease, the plants are able to turn the table on the pathogen and occasionally detect these effectors. And all it takes is one effector to be detected by one of these immune receptors to have a defense response and immune response. I like to use the metaphor of saying that the receptors are like a metal detector in the airport. If the effectors are guns, then if you try to smuggle them through the metal detector, that's pretty hard because they'll be triggering the metal detector. So essentially, that's what plants have. They have a whole bunch of these metal detectors that are designed to detect these effector guns, if you like, and all it takes is for one of these to be activated for the plant to initiate an immune response and defend itself against the pathogen. This is important because plant breeders have been introducing these R genes, as we call them, or resistance genes through classical breeding into plants for decades. They're very useful. There are many of the crops we grow, many of the varieties you grow, many of the added value that seed companies provide is to actually deliver new R genes. But plant breeding, the classical way, is a little bit painstaking. It's a long and slow process. And I'm just going to tell you about a different approach now. So, for example, this is a project that's led by the Two Blades Foundation, working with folks like Marc Ghislain of the International Potato Center, or CIP as it's known from its Spanish acronyms. Also with my colleague Jonathan Jones at the ScienceBee Lab. So what they did here is they took 3 of the R genes, or the disease resistance genes from that were identified from wild potatoes that function against Phytophthora infestans, And they introduced them using the transgenic approach, so using DNA transformation. They introduced them into a potato variety known as Victoria, that's widely grown in East Africa, in particular in Uganda. And this variety, known as 3R Victoria, is really effective, as you can see here. So in the absence of fungicide treatments, this variety is quite immune to the blight disease, the potato blight disease, and therefore is quite favorable for the farmer. Why is this important? Well, it's very important because it's not just for having the better yield in the absence of fungicides. You could always argue that they could use fungicides, but a lot of these potatoes are grown in developing countries, low resource places where the farmers either cannot afford to buy the fungicides, or if they buy the fungicides, they'd be misusing them. This is a picture from a colleague, a professor in Punjab University in India. He sent me this photo. It's really heartbreaking because this poor farmer here, his potato field is blighted. You can see a lot of sick potato plants, and yet he's actually spraying the field. It's totally hopeless at this point. There's no reason why he should be spraying, but he was probably misled into buying fungicides and using them. He's using the wrong nozzle. This is a nozzle for herbicides, not for blight disease. He's not wearing any protection, and so on and so forth. You could actually almost have a sort of a contest here in trying to figure out how many things are being done wrong here. So the poor man is losing his farm and losing his crop, and he's really desperate. So we need to deliver blight-resistant potatoes to farmers like this one. And as someone who grew up in a developing country, I grew up in Tunisia, I know how misused fungicides are in these parts of the world. The farmers often either are not well trained in using the chemicals, or either cannot afford protective equipment, and so on and so forth. So we shouldn't forget that bringing resistance, whether through classical breeding or through modern methods, has direct impact on the farmers' health and the community health, farming community health, besides protecting the crop. Okay, let's switch gears and talk about another modern method that I'm very excited about. I think it has completely transformed biology. This is the CRISPR method. The CRISPR is a method for gene editing. It was very, very quickly, it was only invented in 2012, 2013, so about 10 years ago. And in just a few years, these two wonderful lady scientists, Emmanuelle Charpentier and Jennifer Doudna, received the Nobel Prize because the method just really took biology by storm. And that's why we talk about the CRISPR craze. What is CRISPR? Okay, so I don't want to get technical again, but think about it as a gene editing method which allows you to edit the text of the genome. So, you know, every organism has a genome, which is essentially like a book, literally, with text in it. And what CRISPR is like a word processor. You can go into this book, to any part of the book, any part of the text, and literally cut it and modify it. You can remove sentences, you can remove chapters, or you can even simply remove one letter, which, as far as genetic modification, is the ultimate. Because if you can change one letter by another letter, then you can do better than that. That's like the ultimate in editing, whether it's a word processor or gene editing. And it's very exciting now because it looks like the UK, like many other countries, Japan, Australia, the US, is taking a progressive stance in terms of lightly regulating the gene editing method, because it is truly transformative. So gene editing, we were involved in 2013 in applying it in plants. My colleague Vladimir Nekrasov, who's currently in the Northampton Research Campus, has developed the method, applied it to plants, and has many applications for plant breeding, for accelerating plant breeding, but also doing modifications that are more precise than the classical breeding approach. So how can we apply this to plant health? How can you apply this to these processes I was telling you about? Well, remember how I told you that the pathogen have effectors that are essentially like keys that are unlocking plant processes. So what we can do with CRISPR is go to the lock that is targeted by the key and simply modify it so that the key doesn't work anymore. Or even in some cases, we can simply remove the lock. If you remove the lock, then the pathogen cannot unlock the house with its key. So that's exactly what we can do here. Because pathogen effectors facilitate infection, if we remove the lock, then the key doesn't work and the pathogen cannot infect the plant and act and be virulent on that particular plant. So this is exactly what we did. This was a pilot project that we did years ago with Vlad, and we developed this tomato we call Tomelo, which is resistant to the powdery mildew fungus. And we did that by simply removing these number of letters from the text of the genome. So it was precisely 48 or 49 letters, depending on the plant we generated, that were removed from the genome. So this is interesting because this is the opposite of transformation, where when you make transgenic plants, you actually add genes to the plant genome. In this case, we actually removed DNA or removed part of the gene to make it dysfunctional. And by doing that, the fungus, because the fungus is exploiting this gene to cause disease, then the fungus cannot establish itself on these tomato plants, as you can see in the pictures below. This is important because, first of all, this is the only modification we could detect in the genome of these tomatoes. And second of all, powdery mildew in the UK, in greenhouses, is a problem. It's actually the major reason why UK growers would spray their greenhouse tomatoes with fungicides. My understanding is that the main reason is to control and manage the powdery mildew disease. So delivering this particular tomelo to UK farmers should be quite useful and quite good for reducing the use of these chemicals in agriculture. So here is another use of CRISPR. This is not related to plant health, but this is quite a creative use of CRISPR by my colleague Zach Lipman at the Cold Spring Harbor lab in Long Island, New York. And what Zach did is he introduced 3 mutations in tomato through CRISPR, and these mutations make tomato very bushy, as you can see, and yet quite productive. And in fact, this was done to promote urban gardening of tomato, something that's on vogue at the moment. But the reason I'm showing you this is because Zach actually developed this project of making three mutations, and it's fairly straightforward from a technical point of view. So my colleague, Professor Shao in University of Maryland, he actually was teaching a summer course on the use of CRISPR and introduced Zach's triple mutation in his tomatoes. The students did that. But you know what happened is that summer when they were doing this, I think this was two summers ago, their tomato were bushy, but they got hammered by powdery mildew. Totally hammered. I mean, as you can see here, these are tomato leaves totally covered with the fungus infection. So what happened now is Professor Shao is introducing a 4th mutation, which is our tomelo mutation, and this is really exciting because that shows you how CRISPR can be used to be very creative here. So it's just like classical breeding where you're introducing new traits and new colors through breeding, but this can be done so quickly and so efficiently, and now it's really up to the breeder or the plant gene editor, if you like, to be as creative as they wish. And my vision really is that all these CRISPR produced traits become almost like lines of code where anyone in the world who wants to breed any mutation can actually access them and mix them with other mutations and produce new varieties in any way they wish. But it's quite exciting because in in the next round of summer training that Professor Shah would be given, they'll be introducing the fourth mutation to avoid having their urban tomatoes being hammered by the fungus. I thought that was pretty cool application. So that's all I had. I want to leave time for discussion and questions. Back to the International Year of Plant Health. I hope I tried to convince you how important plant pathogens are, and maybe how interesting they are, and how plants fight back. And I know we've all been distracted by COVID and the coronavirus pandemic, but I'd like to borrow this quote from my colleague Cristobal Wau, who is an expert in weed genetics. And I love this quote because although medicine is very important, medicine can cure you one day. When we get sick, we need medicine, but plants save our lives every day. And so with that, I'd like to stop there and take any question, but I also want to point you to my blog on Medium, where you can read more on plant killers. I try to post regularly on stories related to plant pathology, but there's also a number of other things that might be of interest to you. So check it out on Medium. And so with that, I'll be happy to answer any questions and engage in the Q&A with everyone. Thank you for your attention.
Thank you very much, Sophien. That was a fascinating talk indeed. We have a number of questions in the Q&A, but please, everybody, if you have a question, please put it in the Q&A, and I will pose it to Sophien. So the first question goes back to the first example that you gave, Sophien, and this is relating to the Puccinia monoica infection. Are there additional pollinator cues beside the yellow spore color, for example, UV fluorescence or production of unusual volatiles that attract the pollinators?
Yeah, absolutely. So, okay, first of all, UV fluorescence, I'm not aware that there is UV fluorescence. I haven't seen any papers on UV fluorescence, or I haven't observed it myself on these flowers. But I should mention that this was originally discovered by B.T. Roy, who's at the University of Oregon. When she described this, she made a strong case that these Puccinia flowers are mimicking buttercups that are quite common in that area. So the yellow flowers, the yellow colored flowers are actually quite dominant in that area. And so the fungus may be taking advantage of insects' attraction to the yellow color. But in addition to the color and the morphology, which is also potentially cue for the insects, there's definitely volatile production by the fungus that is absolutely critical. In fact, you can smell these. Next time we'll make a video where we smell them. And it's they have a strong smell. And what the fungus is doing is shutting down the host volatiles. The host produces volatiles through sesquiterpene synthesis, for those of you who know this stuff. And the expression of those terpene synthase is completely zero in the pseudoflowers. It's just totally shut down. But instead it's producing benzaldehyde type, I think it's benzaldehyde, my memory serves me right, type essentially volatiles that are typical of this type of fungi. So yes, absolutely, the fungus is modifying the plant in many different ways.
And does that mimic the scent of any yellow flowers nearby? Because I know that buttercups don't really smell, do they? Or
yeah,
at least to us anyway.
Yeah, I don't think that's been shown or demonstrated. But one thing that's really interesting, if you have a pseudo flower, there's a publication on this that I find absolutely fascinating. The fungus, the attraction to the fungus is so strong that the neighboring plants, even if they're different host plants, different plant species, they tend to be pollinated less because the insect will zoom in on them. So there's a negative impact actually on all the surrounding flowers. So yeah, the fungus is investing a lot into the volatile, into attracting insects. It's not, doesn't face the same type of trade-offs that the plant flowers would have. So it's a really fascinating ecosystem. So if you have a pseudoflower, it's actually messing up with the whole flora of the pollination of the neighboring plants.
That's really fascinating.
Isn't that fascinating? Yeah. So the plants around it would produce less seeds, which is just stunning.
Yeah. Gosh.
Indirect effects.
That's incredible. Rather than carry on myself speculating with other things, I'll pose some more questions because they're coming in rapidly at the moment. The second one, is it possible that adding or removing genes to improve resistance to one pathogen may increase vulnerability to other pathogens?
Yes, Jim. Yeah, I mean, that's, of course, that's always possible. The only thing I can say is that a lot of the resistance genes I was talking about, these R genes, they tend to be quite specific. So they're generally effective against just a few races of a pathogen on one particular pathogen species. So that's with those genes, it's less likely. But with the strategy of removing genes, as I mentioned with tomato, what's really interesting about that, of course, the question that I always get, I'm sure you're thinking about that, why is the gene there in the first place, right? Well, it's very easy to answer that. The gene is there for the tomato when it's growing in the wild. It's not there for domesticated tomato, which is essentially a pampered tomato, right? So it's almost like I actually have usually a picture of a hairless cat. You know, if you're having a domestic cat, you can have a hairless cat if you're allergic to cat hair, and that will grow perfectly fine in your flat. Obviously, it won't survive in the wild, but it would be growing fine and in the pampered environment of your apartment. So so it's the same thing with our crops. I mean, our crops are not growing in the wild, they're pampered, they're being taken care of in an artificial environment. So for example, you could imagine removing all the flowering genes of potato and you still get potatoes, right? So that's a way to think about it. So yes, of course, in the wild, that gene is important, but under domesticated conditions, it needs to be assessed.
And this next question relates to that. By removing genes from the first generation, do those genes just disappear, or do they appear randomly in following generations?
Yeah, that's a good question. So this gene editing is a genetic modification, so it's by definition heritable. So once you remove it in that particular lineage, in the progeny of that particular plant will always carry that mutation. Of course, there would be other varieties of plants that you wouldn't have edited that will still carry the gene. So the gene is not lost for humanity or for the breeders, but it's lost in that particular variety. And
the next one. May there be a time where my garden tomato won't be hit by blight?
Well, if you know, it's I hope so. I would say at the moment there is no. There are some varieties out there that are resistant, and I encourage you to look into some of the catalogs that are out there. There is. This is, you're asking about tomato, but with potato, there is the Sarpo Trust, which is doing the Sarvari Trust. Sorry, you have the Sarpo potatoes, and they're doing a fantastic job breeding resistant potatoes. Unfortunately, the GM technology is not widely used, and so otherwise we can easily provide you with the blight-resistant tomato. But right now we don't have the equivalent of that powdery mildew resistant tomato for blight. So that's something that's still being worked on. So whether in time this will happen and you'll be able to grow it in your garden, Nathan, I don't know. I hope so. But we're working hard on that.
So where in the world are CRISPR manipulated crops
yeah,
legal?
Many countries, pretty much most countries besides the EU at the moment.
EU, yeah.
Right, the EU is still the stumbling block. And but you know what's interesting about CRISPR and gene editing is that countries that are traditionally conservative in their regulatory approach to plant breeding, for example, Japan, which has been traditionally extremely conservative, has actually agreed to lightly regulate CRISPR. In fact, there is a gene-edited tomato that's on the market in Japan right now. So it's mostly Japan, North America, Australia, various other countries. I mean, recently Kenya, for example, has also been positive about it. But the EU position, which is actually being revisited now, is really has wide-reaching impacts. For example, I was in South Africa a few months ago, and South Africa exports a lot of food to Europe and to the UK. I mean, a lot, right? I mean, you go to the supermarket in winter, most of the fruits come from South Africa, but they can't use this technology because the EU market is not open to them. So if you think about it from the perspective of the South African farming community, they would love to use this technology. They would reduce the use of chemicals. It's good for the health or anything, if anything, but they can't do it because they export to Europe, and Europe doesn't want that. So because of the EU position, this is affecting farmers in developing countries, in South Africa, in Kenya, in Peru, where these are countries that export the agricultural produce to Europe. So, so it is a problem. It is really a serious problem.
Yeah. And what about the UK position now that the Brexit has happened?
Yeah, so the UK has, sorry, I'm kind of acting as if everyone knows that, but the UK has agreed to, has engaged in a path to deregulate gene editing, or to lightly regulate it, is the better way to say it, or to regulate it differently from GM and GMOs. It hasn't yet happened. It's a process, but this process has started, and there is a lot of expectations from the plant science community that gene editing will be lightly regulated in the UK in the near future.
Thank you. Here's a question from Nathan. What would you say is currently the most concerning plant pathogen for scientists and farmers in terms of the scale of its impact?
Yeah, I mean, it's always been the blast fungus that's a really huge impact. First, because this is a pathogen that infects cereals, so I can make the pun cereal killer. And this pathogen infects rice and is a major constraint for growing rice in many parts of the world. But more recently, there's a new pandemic of a different strain of the blast fungus, which infects wheat. And this is really scary. This pathogen has first emerged in South America, in Brazil in the 1980s. But just 5 years ago, 6 years ago, it popped up in Bangladesh, in South East Asia, started spreading in the country, is now pretty much throughout the country, possibly moved across the border to India. That's a politically sensitive question to address. But then further, it also appeared in Africa. The same pandemic strain of wheat blast has now popped up in Zambia, and maybe even spreading to neighboring countries. So we're very concerned about the wheat blast pandemic.
Thank you. And now Stephen Runo, he thanks you very much for a nice talk and asks, in benthic infiltrations, does it always work for all effectors? I am thinking of a situation where a specific host is required and it is not in benthic.
Yeah, that's a good point. So it works great for pathogens of benthic and its relatives, which is benthic is a Solanaceae, so it's in the nightshade family. So it's a cousin of potato and tomato and eggplant and so forth, and pepper. So it works great for those pathogens. If you go, for example, to the blast fungus, where it's a pathogen of rice and wheat, and that's really diverse, that's really distant from, phylogenetically distant from benthic, then it doesn't always work, but it can still work when you reconstruct the immune response. So we routinely do experiments where we bring in rice R genes or wheat R genes and then get the immune response to function in Benti. And that's really brilliant, because it's so difficult to work with wheat, for example, and then if you can reconstruct that response. So to study immunity, perhaps less so of studying effectors, but to study immunity, it's still a brilliant system for pretty much any plant pathogen combination.
Yeah. Thank you. And here we are, one from Everton.
Oh, Everton.
Yeah. Dear Professor Sophien, thanks for your excellent talk. Is there an assembly of Puccinia monoica available?
Thank you, Everton. That's Everton is asking about genome assembly. No, there isn't. So there is some genomics data available from way back, but when we worked on it, but no, as far as I know, there is no genome sequence. Maybe you won't sequence it.
And now from Trevor, I grow blight-resistant potatoes on my allotment in Oxford. What is the mechanism of the resistance, e.g. crimson crush?
Yeah, I'm not, thank you, Trevor. I'm not familiar with crimson crush itself, but I know that there are some R genes that were discovered in the US about maybe 20 years ago and have made their way into some of the varieties. So potentially crimson crush carries one of these. So there was a breeder, where was he, in Virginia in the US, and he identified a couple of genes that he combined in his crosses, and that's been now more widely used. So the question here is, okay, it's resistant so far. It's great because it's in a variety that's growing in allotment garden and so on. So potentially there is not a lot of pressure perhaps for the pathogen to overcome it, but the thing is with Phytophthora is so good at overcoming these resistance genes and evolving new races that will overcome them. Because your allotment garden is not a monoculture, and I encourage you to sort of maintain that. Don't encourage your neighbors in the allotment garden to grow crimson crush. That will only increase the likelihood of the pathogen to evolve a new race. So it's a little bit like with COVID, right? We hear about all these vaccine breaking strains, etc. So let's hope that crimson crush will not crush and that the pathogen will not overcome it. But that's a possibility. So don't be too surprised if it breaks down in a year or two.
And now here's quite an interesting question that sort of extends into humans. I guess the question here is are there fungi that have evolved to the point where they're capable of doing similar to what they're doing in plants in humans?
So are we zombies ourselves? Maybe there's maybe I'm infected by a parasite that is manipulating my behavior, and yeah, it's possible. No, but I mean, why don't you Google zombie ants, and then you will see all the crazy things fungi do to ants. And I mean, it's not completely far-fetched, this question. There are actually many parasites and pathogens that manipulate the behavior of animal hosts, including humans. There's even some research out there, I'm not totally sure how robust this is, but that describes toxoplasma, which is the cat-transmitted parasite of humans that is manipulating the behavior of the mammalian hosts, like mouse and maybe humans, in terms of making them act in a more risky way. So all of a sudden, a mouse that's infected with toxoplasma is coming out in bright daylight and running around in the open, whereas normally mice never do that. And this increases the likelihood that they get caught by a cat, which is the host that the toxoplasma really wants to get in.
Oh.
Yeah, so it's pretty fascinating. And there are some studies showing that toxoplasma infected humans are more likely to get speeding tickets and things like that. So I don't know how solid this is, but there are some theories out there. I wouldn't be shocked that there are parasites that affect the behavior of animals, including humans. So. But again, check the zombie ants. They're quite impressive. You see what fungi can do to animals.
Yeah. And the next question, again, relating to CRISPR, can gene editing through methods like CRISPR, actually weaken the usual plant resistance to a variety of pathogens that you mentioned?
Yeah, I mean, this question is very general because what's underpinning this question is, I think, a confusion that's very common in the discussion about these methodologies. There is the method, and then there is the product. Obviously, you can use the method to generate any kind of mutation in the plant, which could even cripple it completely. But so does mutagenesis, or so does classical breeding. You can actually introduce toxins by breeding wild potatoes with cultivated potatoes, or even the tomatoes we eat. You know, many of us are actually allergic to the leaves and allergic to many tomato compounds. So even classical breeding can exacerbate these problems. The point is that there's nothing with the gene editing method per se that makes it more likely to weaken plant resistance or even to increase plant resistance. It's just another method to introduce genetic modification. Then once the genetic modification is introduced, independently of the method, it could be good or bad, right? So I think we need to separate in this discussion the process that leads to the modification and the modified plant itself, which obviously needs to be studied and goes, even classical varieties, if I breed a classical variety of tomato, I still need to take it through a regulatory process to make sure that it doesn't have toxins and these sort of things. So yeah, theoretically anything can happen with genetic modification, but it's not specific to the method.
Thank you. And I'm conscious of the time. It's just gone 8 o'clock, but we still have some questions. Perhaps we can.
It's fine. I don't mind going on.
Race through these. Okay. Here's another one from an anonymous attendee, or the same anonymous attendee. Is this potato pathogen active where potatoes are originally from, i.e. South America? Was it brought with them?
Yeah, great question. I mean, that would have been another talk. I can talk about that because we actually did sequence the genome of the strains of the potato blight pathogen that are in Kew Gardens that date back to the 1840s from Ireland. So we know exactly what sort of pathogen came to Ireland. So when potato was introduced to Europe for a couple of centuries, there was no blight. So blight came later, and actually the pathogen came from Mexico, and it was introduced to Europe in the 1840s, most likely through sorry Simon, but most likely through Botanic gardens.
Yes,
from wild potatoes that were brought to Belgium, I think, through the Botanic garden there, and then it spread to Europe. So the potato came before the pathogen, and the potato came from the Andes, but the pathogen came from Mexico, from Central America.
And the next one, again, thanking you for your excellent, accessible talk. I wonder why the plant genes that the Phytophthora effectors target have persisted in the plant over evolutionary time. How is it possible to just knock them out without side effects?
Very, very good question. So the first part of your question is why these genes persisted is because they're probably important. They must be important for some plant processes, right? But that doesn't mean there's no coevolution going on, because even though the gene is there, there are still mutations that will arise in the gene to evade being detected by. So it's the changing the lock metaphor, if you like, right? So the plants are also continuously changing the lock naturally, changing the nature of the lock, so the key doesn't work. Then the pathogen gets another key or modifies its key. So that coevolution is going on all the time. That's an arms race that's going on between the receptors and the effector they detect, and between the locks and the keys, if you like. So why we can modify them without side effects? So I think I answered that question earlier. So because domesticated plants are pampered, so we are modifying genes that are not important for growing our crops under agricultural conditions. They probably would need those genes and in the wild, but when we grow them in our pampered farms, then or allotment gardens, then they don't need that gene.
Right, and the next question relates to
Oh,
this Red Queen arms race. How fast do you think the fungus might develop a way to elude the resistance gene?
That's the question. I mean,
Engineered.
really,
Yeah.
that's the key question: durability of resistance. How can we make this resistance durable? And all I can say, I mean, I like to say that never bet against the pathogen. My view is the pathogens will always evolve a way to figuring out how to overcome the resistance. So it's really a question of time. Can you make a resistance that will last for 10 years, 20 years, which would be useful enough before the pathogen evolves new race? A little bit like what we're dealing with the vaccines and the coronavirus vaccines. You know, like now there's a new formulation. You know, I'm scheduled to have a shot in a few weeks with the Omicron, with the sort of bivalent version of the vaccine. So it's a little bit like that. The idea is really to delay the evolution of the fungus by maybe combining, for example, multiple genes. So if you have 3 or 4 of these together, that's why that 3R Victoria potato has 3R genes together. That will make it harder for the pathogen to evolve. So that's the sort of strategies we are using to, and also reducing monocropping, because the more uniform the crop is, the easier for the pathogen, the higher the selection pressure will be for the pathogen to evolve. It's a number game. It's there's a lot of math involved there. Maybe some of you are mathematicians. You probably can model this better than I would.
Yes, which takes us back to sort of medieval times when cereals were not grown in monocultures, but were grown in sort of biodiverse populations, so that one in a bad year for pathogens, some would survive and some wouldn't. And similarly with droughts and conditions like that. And some farmers are going back to this sort of biodiversity population farming. I know some near Oxford that are doing that with various cereals. And that sort of brings us into this question about regenerative farming. And it says regenerative farming approach to growing healthy soil uses no additives or pathogen controls. Is the potato pathogen less successful against healthy soil and healthy plants?
I'll be honest with you, the potato blight is a really nasty disease. That's a very aggressive disease, and generally the agricultural practices have little impact on the potato blight, as I think many of you know from growing potatoes and tomatoes near allotment gardens. If you don't have a resistant variety, it's really hard to manage it without chemicals, right? So yes, of course, it can make a difference in terms of the intensity of the disease, but it's very hard to completely get a fully immune plant, and it's just so aggressive. When I think again, all of you know it, that when you have the right conditions, maybe we're not getting these conditions anymore as frequently as in the past, but when you have those cool and humid conditions in the summer, and you really have to worry about your potatoes and tomatoes here in the UK and most of the world actually. So it does make a difference, but there is a difference between quantitative effects and then the qualitative effects that plant resistance can confer, which is total immunity.
Okay, thank you. Now, I'm going to have to draw things to a close now, mainly because my battery is showing that I'm running really low, and I want to thank you so much, Sophien, very much for a fascinating talk this evening, and great opening to our autumn lecture series. The next one will be in 2 weeks' time with Phil Poole from the Department of Biology here at Oxford. So a big virtual clap from everyone to you, Sophien. Thank you so much for a wonderful lecture. And good night, everybody, and look forward to seeing you all back here in 2 weeks. Good night.
Thank you.
Good night.
Good night. Thanks.